Upstate Medical University in Syracuse, New York invites you to be the informed patient with the podcast that features experts from Central New York's only academic medical center. I'm your host, Amber Smith. You may have heard of the protein tau that plays a role in Alzheimer's disease. Today, I'm going to talk with a computational neuroscientist from Upstate about tau seeds and their role in this disease. Chris Gaiteri is an associate professor of psychiatry and behavioral sciences at Upstate Medical University. Welcome back to The Informed Patient, Dr. Gaiteri.
Hi, Amber. I'm so happy to talk to you about this.
Well, before we get into these seeds, can you first tell us about tau, what tau is?
Sure. Tau is one of the two proteins that you need to have in your brain to be classified as someone who has Alzheimer's disease. It was discovered a long time ago because it tends to aggregate and it's visible on a microscope when you're looking at the brains of people with Alzheimer's disease. And so I can tell you about tau, but it has a twin, not a twin, but a sibling called amyloid. And I can't really talk to you about tau without also talking about amyloid. In fact, there's sort of been this historical rivalry between tau and amyloid scientists in terms of which one of these is the most important in producing the symptoms of Alzheimer's disease.
So they work together, or do they oppose one another?
They don't oppose one another, unfortunately. If they did, that would probably be helpful. But there's been a debate about which one of them comes first, which one of them is most related to the cognitive symptoms that we really care about. So initially, the pendulum sort of swung more toward amyloid because there's a special kind of Alzheimer's disease called familial Alzheimer's disease. And it's unfortunately a very severe form of the disease, but there's a genetic variant that drives the disease, and it's in this amyloid precursor protein. And so that encouraged a lot of people to believe that this was essentially the core of the disease. But now that we have anti-amyloid drugs that are unfortunately not that effective, people have begun to consider if tau might be more important. And indeed, tau begins to aggregate and sort of build up in the brain at a time much more closely to when people are beginning to experience symptoms. So amyloid, you can see it beginning to build up 15, 20 years ahead of the disease. And so people were attracted to it initially because it looked like it was a very early marker of the disease. And indeed, there may be value to knocking it down. But with tau, it's rising much more closer, a few years, let's say, ahead of a really noticeable cognitive decline. So now people are investigating tau perhaps more seriously than they did 10 or 15 years ago.
So a healthy person who doesn't have any cognitive decline doesn't have amyloid or tau. These don't exist, right, naturally, unless they're heading toward a disease.
That's actually a really interesting thing. You would think, based on everything that I just said, that they wouldn't, but actually they could. Not necessarily, but there's about a third of older people, say 65 plus, who have amyloid and tau in their brains at levels where we can easily detect it. And cognitively, they're actually fine. So it's not like it's a sine qua non of Alzheimer's disease. It's a heavily predisposing factor, but it's not a guarantee that you're going to have the cognitive symptoms that we really care about.
Wow, that's interesting. So what does tau do, or what does amyloid do in the brain? Are they destroying parts of it, or what is their presence doing?
Yeah, so actually you need tau to develop normally. And you also need it in your early adult life because what tau's doing is it's helping with molecular transport. So if you think about a neuron, you've got a sort of a main cell body, and then there's a long extension out of that called an axon, and that's how it's transmitting information. You need to get proteins down this narrow axon, though. And so there's actually these things called microtubules that are basically rods that are supporting this long highway towards the end of the cell, towards the end of the axon. And tau basically is holding those rods together. And that's important because there's yet other molecules that grab onto proteins and will sort of walk one molecular foot after the other down these rods, dragging the proteins along with them. And so tau is kind of stabilizing this whole assembly, this whole transport mechanism that is essential to moving proteins where you need them to be. So It absolutely has a positive function that you need to survive. But there are certain forms of tau, certain molecular shapes that it can get stuck in that are definitely problematic. But to your question, that's what tau is actually doing, is it's stabilizing basically these highways of the cell that you use to drag proteins where they need to be.
Now, what are neurofibrillary tangles or fibrillary? I'm not sure how you say it.
I go with fibrillary, but if you said fibrillary, that would be fine too. So these are collections of the tau protein, often with a bunch of phosphates attached to them. And you're going to find these inside the cells. Amyloid outside the cell generally. These are inside the cell though. And it's basically this long snaking collection of the tau molecules that's zigzagging and swirling all inside the cell. And in that form, in that way, where it's basically this long snake inside of the cell, that interferes with molecular transport, which is in fact the very thing that it's sort of there to do in the first place. So it's somewhat ironic. Yes, those are the neurofibrillary tangles, those long snaking lines of tau inside the cell.
This is Upstate's The Informed Patient podcast. I'm your host, Amber Smith. I'm talking with Upstate computational neuroscientist Chris Gaiteri about tau seeds and their role in Alzheimer's disease. So let's get into what are the tau seeds?
So it's called a seed because it's the beginning of something much bigger, like an actual seed that you would find out in nature. It could be even a single molecule of tau that's just misfolded in a certain way that when it bumps into another molecule of tau, it also becomes misfolded in a certain way. And when those two bump into a third molecule, that one also becomes misfolded. And when I say misfolded, I essentially the protein adopts a shape that it's not supposed to have if you're healthy. And so this is sometimes called a prion-like process. There's other prion diseases where we see this with tau or with other proteins where basically they misfold, they cause other copies of themselves to misfold, and it just goes on and on and on. And then they'll fragment, break apart, and they'll cause other molecules to misfold. So to your question, that's why it's called a seed, because we think that the tau seeds are essentially molecules that are at the very beginning of this really nasty process.
Are all of the tau seeds misfolded, though?
It's sort of the definition that if they aren't misfolded, they're going to be soon, but basically all of them. When we're talking about a tau seed, it's kind of the definition of this is the thing that's going to cause other things to misfold. So yes.
And this is going to sound like a silly question, is it a physical collision that causes them to misfold?
I wouldn't call it a collision, but it's a complex attraction between the two molecules. They have some affinity for each other. And once they essentially come into contact with each other, not violently, but they just sort of come into contact with each other, the one molecule will cause the other molecule to
Once they're misfolded, what do they do? Is there any function, or are they in a state that they can't do anything after they've misfolded?
Unfortunately, yes. At that point, they lose that positive function. They're not doing their job of stabilizing these highways that allow molecular transport. As far as we can tell, they're not doing anything good at that stage. And that's the beginning of these long fibrils that are just sort of going to snake through your neurons and cause problems. So yeah, once you're at that stage, it's
Once they are misfolded, is there any way to straighten them out again?
That's a really good question. There are a whole bunch of protein clearance proteins, actually proteins operating on other proteins that clear misfolded proteins out of the cell. So you do have molecules, every cell in the body does have these molecules to essentially clear out these misfolded proteins. So that would be potentially, one mechanism to dealing with these. There's also clinical trials for anti-tau antibody drugs. So these also would be essentially breaking up tau and clearing it out of your brain. So there are ways of dealing with it. Yes.
But the tau seeds, they stay in the brain. They don't go throughout the body, right?
You would think, but actually, I have friends in Sweden who measure the CSF, so the cerebral spinal fluid, which is a fluid that circulates through your brain and then down into your spinal cord. And so it's a kind of a way to get access to or a peek at what's happening to your brain. And so they've sampled the CSF of a few thousand people there, and they've shown that they can find misfolded tau in the cerebrospinal fluid in the CSF. And so it makes it that far. And then I have some other friends in Chicago who have been measuring it in the blood of some of these individuals that we've been studying. So you can actually see the misfolded tau showing up specifically, not just tau. So your brain is unfortunately generating a lot of it because of this prion-like process, because of this propagation that the seeds do. And it does actually end up leaking out into at least the blood and your CSF.
Now, these tau seeds, they're linked to Alzheimer's. Are they linked to other diseases as well?
They are. What disease you end up with depends on a few things. So it depends on what cell type the tau is in, where in the brain the tau is located. It also depends on exactly what version of the protein you have a lot of. So you can have tau in chronic traumatic encephalopathy, CTE, the disease that is a problem for a lot of football players and boxers. Pick's disease is another tau disease where it's in a different spot. There's a variety, frontotemporal dementia, there's a variety of diseases that seem to have a strong influence of tau and probably tau seeds. And it's really just a matter of where the tau is located in the brain. You will get certain symptoms.
Now, I know you're a researcher, you're not a clinician taking care of patients, but you mentioned that you have some colleagues that have found these tau seeds in blood. Could a patient have a blood test, maybe not today, but in the future, and determine whether they have tau seeds?
Yes, actually, you could. You could do that. You could actually look at a whole range of forms of tau. So basically, the tau molecule can have phosphate groups stuck on it at different locations, and where they're attached, how many of them are, could be indicative of your future health. So yes, this could come out of the research setting and become a test one could have. I think it's likely that it would need to be used in conjunction with a bunch of other tests as well, because it doesn't have the sort of clear predictive certainty. It's not going to tell you with 100% accuracy or even 90% accuracy that in 5 years you're going to be having certain symptoms. It's really not at that level. It's more of a research tool at this point. You could have a blood test.
Would the tau seed show up in a brain scan?
Not directly. To see tau in the brain, you actually need a specific form of tau, which is not the seed form that's going to be lighting up on a PET scan. So you can infer, though, that if your brain is loaded with tau, which you can see on certain brain scans, you probably also have a seed problem. One thing that we've been doing is sort of tracking the process of the seed unfurling into tau. And you're sort of asking, in a way, about the reverse issue, which is if you know that you have a bunch of tau in the brain, which you can find from certain brain scans, do you have the seeds? And so it's you can infer that you do, but I would say those brain scans also are mainly a research tool. I'm not even sure if there's one available in the Syracuse area.
Now, does the accumulation of tau or does the development of tau seeds cause any symptoms in a person?
Yes, it does. They're going to be increasing the likelihood, and this gets to some of our recent paper, but if you have tau seeds that are active in a certain area, they're going to be increasing amount of neurofibrillary tangles in that region. If you have increased neurofibrillary tangles in certain brain regions, that does increase your odds of having Alzheimer's disease. That does increase your odds of having various cognitive problems. So, yes.
Now, for the study you were involved in that was published in the journal Neuron, you were working with brain tissue donated from nuns. Is that right?
Yes. Nuns were wonderful participants in this study, and also priests, brothers and sisters, and non-clergy too. These are generally people from the greater Chicago area, and they all promise to donate their bodies to science after they die. But before then, every year they come in, or we go to them and give them cognitive tests. We take a whole bunch of measurements. We scan their brain. We have blood draws. So these are actually literally the most measured people on earth, especially when you consider all of the brain data that we have from their brain after they die. We measure the levels of genes like RNA. We measure protein levels. We measure the level of different molecules that are attached to different spots on their DNA called methylation, acetylation. There's actually billions of measurements that we take on these people.
So were you able to determine the amount of tau and the extent of tau seed activity?
Yes, we did. And it's pretty cool because what we actually do, and when I say we, there's collaborators at the University of Alabama and Rush University who are doing this, and they do a fantastic job. You actually take an extract of a person's brain from a specific region, and then you stick it into an immortal human kidney cell, and then you measure the fluorescence of the tau because you have this other molecule that attaches to the tau seed and then fluoresces. And so you can actually, that's what it takes to actually measure this thing. And so you have a measure of how much of that tau seed you have through this kind of exogenous assay, through this sort of like, in vitro dish assay. So we measure the tau seeds, and then that's really the basis for understanding everything that happens after that. That's essentially what this study is trying to do, is say, given that this person has this many or this amount of tau seeding in a certain area of their brain, what's going to happen after that? What does that entail for this person? That's kind of the question we were asking in that study.
In the subjects that had a lot of tau seeds, did they all have Alzheimer's? I mean, has it been established that tau seeds are connected to Alzheimer's?
There's a definite prevalence of Alzheimer's disease in these individuals. In the people that we were studying in the study, about half of them had Alzheimer's disease, maybe a quarter had mild cognitive impairment, and a quarter of them were cognitively healthy. And as we talked about before, just being cognitively healthy doesn't mean that you don't have amyloid and tau in your brain. You may well have it, but they were at least cognitively doing well.
So what else did you learn about tau seeds?
Well, I think the coolest thing from this study, maybe not the most important, but the coolest thing was how we had people who were doing microbiology, electron microscopy, like examining these really minute structures in a cell, working together with people who do neuroimaging, which is at the other end of the spectrum, right? You have living people and we're studying their brain activity. So you're kind of uniting all of these different scientific activities together. And what we basically found was we could trace the activity of the tau seed from this molecular level, where you have a tau seed, which is a single, but in many ways related to a single protein with certain phosphate groups attached to it, all the way up to saying that tau seed tends to transmit more from early Alzheimer's regions to later Alzheimer's regions based on how correlated the brain activity of those two regions is. So let me put it another way. We measured tau seeds in multiple brain regions, and we can show that based on, we call the functional connectivity between two brain regions, which is basically how correlated their activity is, you would be more likely to essentially have tau seeds and tau itself spreading from one brain region to another. So basically, it's a really unfortunate situation where the brain's own correlated activity, which is really essential in driving our cognitive life, tau is kind of, in a sense, hijacking that and using those relationships between brain regions, which of course are driven by neurons that are connected to other neurons. And so you have this seed that is essentially propagating along those physical pathways. And that may be very important in understanding how tau ends up where it ends up.
And that would decide which type of, like there's frontal temporal lobe dementia that affects the frontal temporal lobes, but there's other dementias that affect other parts of the brain, right?
Indeed. And one thing that we did was we actually combined genetics with tau levels. So we can measure the genetic variance that each person has in this data set, along with how much tau seed they have in a particular region. And we're trying to understand, how does that tau seeding plus their genetics predict how much tau is going to spread within a region, and then how much tau is going to spread to other regions that are connected to that core region. So we did this between a region called inferior temporal gyrus. So this is sort of if you point at your head, sort of at the rear of your ear, that's about where that is. And then we're also looking at how that relates to essentially premotor cortex, which is sort of more up at the top of your head. And so these are two regions that do communicate with each other. We can, in fact, measure that communication between the two regions. And we can measure tau in both of those regions. And so we essentially, by incorporating genetics, which doesn't change, we can actually talk about causality, about how tau in one region in the inferior temporal gyrus is actually causally generating tau levels in another region of the brain. So it's always nice to have causality in a scientific study, because a lot of times you'll find these associations, right? You'll hear a report, A is associated with B, but you don't know if A is causing B or is B causing A, or is there some other factor C, which is causing both of them, right? So by incorporating the genetics into this data, we really helped us to develop a causal narrative for how this is happening. And it's not just an association that we found, it's actually causal.
You're listening to Upstate's The Informed Patient podcast. I'm your host, Amber Smith. I'm talking with Upstate computational neuroscientist Chris Gaiteri about tau seeds and their role in Alzheimer's disease. And you're working with human brains. Does this happen in animals?
There's a long and fraught history of trying to simulate Alzheimer's disease in a mouse. And in a way, it wouldn't be that bad to be a mouse, because we have cured Alzheimer's disease in a mouse so many times. So I am really hesitant to make predictions from human to mouse, mouse to human, unless it's like a very specific question. It's not to say that mice are not tremendously helpful and useful and necessary, but I really don't want to make any sort of broad statements about, is this happening in a particular mouse? The other thing is, there's so many types of mice, where we have all different types of mice where we've actually induced human genetic variants that are related to Alzheimer's disease. So it's kind of hard to say in general, does this happen in a mouse too? And that's really been a challenge for Alzheimer's disease, because if we had a system where if we cured Alzheimer's disease in that system, it guaranteed that we were also curing it in a human, we could just go to town on that system. We could try every drug that we have, absolutely everything possible. We could edit it with CRISPR, and then we could find a solution very quickly. But unfortunately, we just don't have that system. And in fact, the better the system is, the harder it is to use, right? I mean, in many ways, monkeys might be a good organism for testing Alzheimer's drugs, but they take forever to develop, right? So that really slows you down. Or so you could be like, okay, I'm going to try to develop an Alzheimer's drug in C. elegans in a worm, right? I mean, they have a lifespan of a few days, so you can test stuff really rapidly in them. But then it tends to be less relevant to humans. So you're always trying to find that sweet spot. And that's really been a big challenge for the field. And so that's why I'm uncomfortable saying what's going on in a mouse.
Well, what about cows? I'm thinking of the mad cow disease that involves prions in the brain, right?
Right. It does. Creutzfeldt-Jakob disease is another prion disease. You might be able to do it there. I think if you're just looking at the propagation from one cell to another, that you can actually do fairly well in a dish. I would use actual human brain cells. So we have a variety of stem cell lines from these participants as well, about over 100 of them actually. So you'll have cells growing in a dish, neurons, and you can essentially put a little bit of tau on one cell and then see how it propagates to connected cells in a dish. And so I think for me, that's what I would suggest as sort of the sweet spot of, it's an experimental model that has relatively fast turnaround time, but it's still human relevant because they are in fact human cells. They are from a specific individual. And then we have other collaborators at Harvard who were showing that actually tau levels and amyloid levels in these cells, even though they're a stem cell basically that's drawn from a person, so it's very different from the person itself, have amyloid and tau levels that are related to the donor. So that's sort of something that seems to be inherited into these stem cells. So that would be my suggestion for where to go if you're trying to study this.
Do you see potential in targeting tau seeds as a way to impact the development or the progression of cognitive decline or Alzheimer's?
Yes, there are some drugs that are in clinical trials at the moment that we suspect are actually affecting tau seeds. Whether they affect tau or tau seeds, it's still sort of being hashed out. But yeah, there are drugs that are addressing that. We all want solutions as fast as possible, so I certainly hope that those are successful. There's even some existing drugs. I think suvorexant, which is a drug that is given for insomnia, there's some indications, not in clinical trials, but just sort of like preliminary indications that may be lowering tau levels as well. So there's drugs out there that are fairly safe that could be addressing this as well. And that's sometimes preferable because essentially you don't have to go through as much of the regulatory approval process. They've already been tested for safety.
Now, at the beginning, you mentioned amyloid as, that plays a role along with tau. Does amyloid affect the seeds in any way?
That's a really good question. There are a variety of things that can happen to tau that essentially turn it into the sort of this monster that's misfolding and causing other tau to misfold. And so there's things like chaperone molecules, which are sort of what it sounds like, sort of like a parrot at a ball. There's molecules that help other molecules fold properly. Maybe there's a problem with those. And so amyloid could be related to that. Amyloid could influence levels of oxidative stress in the brain. Oxidative stress then can cause tau to misfold. So there are ways that the effects of amyloid could be essentially driving effects of.
Well, it sounds like there's a lot to keep you busy researching in this area for years or decades to come.
Well, I would like to wrap this up quickly. Of course, the research side is not this interview, but I would like to wrap this up into some solutions for patients. That's of course the goal. I don't want this to go on for decades. It might, but I really don't want it to. And so we're trying to move fast when we find molecules that we think are causally driving the disease.
Well, Dr. Gaiteri, I really appreciate you making time to tell us about your work.
Always a pleasure to speak with you. Thank you so much.
My guest has been computational neuroscientist Chris Gaiteri, an associate professor of psychiatry and behavioral science at Upstate. The Informed Patient is a podcast covering health, science, and medicine, brought to you by Upstate Medical University in Syracuse, New York, and produced by Jim Howe, with sound engineering by Bill Brokel and graphic design by Dan Cameron. Find our archive of previous episodes at upstate.edu/informed. If you enjoyed this episode, please invite a friend to listen. You can also rate and review the Informed Patient podcast on Apple Podcasts, YouTube, or wherever you tune in. This is your host, Amber Smith, thanking you for listening.